So, physical growth characteristics, exercise 9, and pure culture, exercise 8, kind of go hand in hand. Because if you think about pure culture, the whole reason that you wanted to do pure culture was so that you could isolate one single bacteria. When we talk about the physical growth characteristics, we're talking about what does that colony look like? Now, remember that at the end of the semester, you are going to have to identify an unknown bacteria. I'm going to give you all of this information, and you have to perform tests. If you're face to face, you're actually going to hands on do all of this. But if you're online, I will give you the results of all of the tests, and you're going to have to become a detective who looks at everything and every characteristic that you see. And with that analysis comes to uneducated conclusion, English, of which bacteria you actually have. So with physical growth characteristics, you're looking for those clues. You're looking to see, okay, what does this bacteria look like when it's on a plate? Normally, you will have 12 different bacteria on a plate that have very different looks to each of their colonies. In this case, you're looking for specific growth characteristics. Is it elevated? Is it not elevated? Does it have a smooth edge? Does it have a scalloped edge? What does it actually look like when you see it on a plate? Some of them look very unique. One of them, one of the 12 in nutrient agar, turns pink. When that happens, when you see that, you know exactly which bacteria you're looking at. There's no if, ands, or buts. There's another one that it kind of turns a greenish yellow. Again, if you look at a plate and you look at all 12 examples, and those 12 examples don't look any different from each other, that may be a problem. But if you have one that has the one standout characteristic, it'll be very easy to go, that's what that is. But again, you have to be able to observe as any type of scientist. Observe and record what you see. What does it look like? What do you see? You have to be able to do that. I'm going to show you this slide again. Observation of physical characteristics of bacterial cultures helps to obtain information useful in distinguishing between different strains of bacteria. If I were to look at this, just plain Jane look at it, I would say that there are probably at least 5 different types of bacteria on here. Why do I say that? The colors is one. I've got beige, I've got yellow, and I've got orange, right? But if you look at the colors, this orange and that orange are slightly different. So does that mean it's a different type of bacteria? Not sure yet, but I see that, I can observe that. Also, look at the edges of these colonies. This one is very smooth. This one is fairly smooth. This one is very smooth. Look at this one. It's very scalloped. That looks completely different than these others. So this might be different. This is a different characteristic. This is something that is observable. Now, I will admit, looking at this right now, this is a lot to take in with different cultures. If I were to make my pure culture, I would make all of these observations about that pure culture and the individual colony that I am seeing. Does it have a smooth edge? Does it have a color? In some cases, does it have a particular smell? because with these bacteria, some of them smell really bad, some of them don't smell like anything. So that's kind of another thing. Now, you're not going to take it and go, because yeah, it's bacteria. It's not a good idea. But if you open it and there's an odor emanating from it, that's important to write down. Again, you're a scientist. All of these observations will help you later on. In your lab manual, in exercise 9, so if you look here, you've got the forms of the colonies. Is it circular? Does it look like a bullseye? Basically meaning that it is concentric, which I will show you an example of in just a minute. Is it spindle? Basically meaning, is it almond shaped? Spindle just means almond shaped. Is it irregular? Is it rhizoid? Or is it filamentous? Filamentous, I always think of mold, like bread mold. It looks furry. What do the margins look like? Are they entire? Are they lobate? Are they scalloped? That original slide that I showed you here, I would say that's scalloped. It's got that kind of scallopy edge to it. Is it undulate? Does it look more like waves than it looks like scallops? Is it eros? Basically, does it look spiky? Or again, is it filamentous? Does it look furry? The elevations. Now, this is looking at it from the side. So you're not looking straight down anymore. You're actually looking at it from the side. Is it subsurface? Did it dig into the auger? Did it create a divot? Is it flat? Does it look like it's just kind of sitting there very, very close to the surface? Is it raised? You can kind of see it's above the surface, but not by much. Is it convex, where you can kind of see that it's higher than it would be if it was just raised? Is it pulvinate, where you actually have this huge lump of bacteria? Or is it umbate, where you have kind of a raised center portion, but the rest is just kind of convex? Each of these characteristics may be different for each bacteria. Looking at this one right here, it is circular. That's the form. The edge, the margin, is entire, right? You don't see any scallops, you don't see any spikes. They're all entire. And as far as the elevation goes, if you look at this one, you can see that this is very high. Now, let's look at some of the other things that we might see. So remember I said with filamentous, it looks furry or fuzzy. See this? These would be, well, this would be filamentous. It looks furry. Whereas rhizoid, this one here is more solid. See this? I would consider this rhizoid. It's not furry, but it does have kind of that weird shape to it. Now, remember I said color is important? Look at these. These are green. And this is nutrient agar. And if you look at them, they're definitely going to be pulvinate, okay? Or even convex. They've got that shape to them. Now, this is a mess, but if you look at the individual colonies, actually, you know what? I would say that's probably more umbonate because they've got that kind of peak in the middle. See that circular peak? I also mentioned the concentric up here at the top, right? Look at that one. Doesn't get more concentric than that. It looks like a bullseye. All of these individual characteristics can be useful to you when you are looking at bacterial cultures, especially if you are looking at bacterial cultures that you have to be able to identify. If you can get isolated bacteria make a pure culture plate and get individual colonies, even though you know it's a pure culture. But if you can do that and get individual colonies and basically write down the characteristics for that particular bacteria. What do the edges look like? What does the colony look like? What's the rise? If you can do that effectively, then you have a booklet of data that is actually a lot more beneficial to you than just having a book that you've never seen before. As a scientist, we do tend to use reference materials, but hands on experience is always going to be, I guess, more educational because you actually see it yourself, you do it yourself. Now, I know right now with COVID and everything else, it's practically impossible to do. Don't get me wrong. But if you're really planning on going into science, these are things that are helpful to, well, will be helpful to you when you get into working in a lab. I've worked in 5 labs, I think. And every lab is different. And what I worked with in every lab was completely different.